Serotonin signaling via HDR2B has a dual function, which is dependent on the microenvironment, specifically nutrient depletion lowering survival, and in collagen 1 accumulation increasing the invasion and the epithelial-mesenchymal transition. We see that HDR2B is regulated by the mTOR pathway, and that HDR2B and NOTCH3 have overlapping expression patterns, which together will modulate the invasion potential in collagen. And once again, as a summarizing figure, in suboptimal physiologically stressful conditions, stimulation with HDR2B further leads to cell death, to lower survival. And when the ECM has stromal accumulation of either fibroblasts or increased collagen, we see that HDR2B activation further increases this invasive partial EMT phenotype instead. And as our take-home message, our data provides an explanation to the previously observed contradictory role of CRC, suggesting that the role of this actual receptor is dependent upon the microenvironment and the exact stages of the tumorigenesis. I would like to thank the department in general, all members, and more specifically Professor Uzac for giving me a place in the department, and Dr. Zoltan Wiener for bringing me into his group, and all present and past members of our department. And of course, the Ujuki teaching hospital staff who provided us with the samples. Thank you for your attention.
Thank you so much for the nice summary. We will continue with the opinions. I would like to ask Kristina Takacs first to read her short version with the questions, and you can continue with direct answers to the questions one by one.
Okay. So I tried to shorten reading my evaluation as much as I can. As we heard actually in the introduction, CRC is the second third most frequent cancer type in the developed regions of the world with high morbidity and mortality rates and with significant inter- and intra-patient heterogeneity. Understanding signaling events and tumor microenvironment interactions that potentially contribute to tumor heterogeneity is essential to develop more efficient therapies. Thus, the topic of the thesis is important and timely, and the results presented here might have an importance in novel treatment strategies in colorectal cancer. The structure of the thesis is traditional. The candidate provided a compact, informative summary of the work. The introduction section summarizes critical signaling pathways in colorectal cancer, as we just heard, and gives an overview about morphological and molecular aspects of CRC tumorigenesis, followed by CRC classifications based on morphological, histological, and molecular criteria. In addition, the candidate describes alternative CRC classifiers, the role and composition of tumor microenvironment, and finally outlines the organoid technology. The literature review is illustrated with Idan Karmi's own figures, which are informative, beautifully crafted, and help understanding. I think we saw some here also. Figure 3 was especially useful to me about the consensus molecular subtypes of CRC, CMS1 to CMS4. The objectives clearly explains the aims of the work and the rationale behind the experiments. The materials and methods section presents the applied methodology in sufficient details and discusses the challenges, and we just saw the graphical summary about the workflow, which was, in my mind, very creative. The results section is the role and well organized, clearly explains and interprets the experimental findings. The figures are of high technical quality with informative captions and clear data presentation. The discussion and conclusion sections are cautious and balanced, reflecting awareness of both strengths and limitations of the study. In addition, further experiments and potential extensions of the project are also suggested. I just listed some details about the references. It was also very well balanced between primary research articles and review sources, and approximately 70% of the references published within the last 5 years were used. The thesis meets the criteria of a PhD thesis regarding both format and content. The dissertation is very well written in English, the genetic terminology is properly used. I found almost no typographical or grammatical errors in the text. Then I listed the results that I don't want to repeat now, but I have a question that I would like to ask from the candidate, and just a summary that the thesis is based on 3 original research papers. One of them was published with the first authorship of Idan in the prestigious journal Cell Communication Signaling with impact factor 8.9, and the other 2 publications were published with the co-authorship of the candidate. So we heard that the cumulative impact factor is almost 24, which exceeds the expectations of the doctoral school of Semmelweis University. So I evaluated the thesis of Idan with excellent grades, and now I would like to ask the question. So the serotonin receptor HDR2B mTOR C1 axis emerges as a potential vulnerability under stress conditions, where targeting either pathway may disrupt tumor cell adaptation and improve chemotherapeutic efficacy. Could be targeting of both pathways simultaneously result in a more efficient way, therapeutic way to treat CMS4 type CRC? Although we know that already mTOR inhibitors have a lot of side effects, but still, can you imagine this strategy as a more efficient strategy?
I can answer.
Sure, please go.
So, thank you very much for your wonderful review, and to answer directly your question, in the literature already there have been demonstrations of dual active pathways in which, not particularly this one, but in other cases where you target both the metabolic pathway of the cancer development and also different GPCR activities simultaneously, and those have been proven to be somewhat successful in preclinical and initial clinical studies demonstrated in the literature. And so this is initially already a great possibility. Additionally, we see that there is, to a certain extent, almost a loop in terms of the way that HDR2B and mTOR interact with each other. As I put on an early slide demonstrating how the HDR2B activity works, we saw that mTOR was downstream to it. However, on a later slide in my results, I demonstrated that when treating with rapamycin, an inhibitor of the mTOR, HDR2B decreased. And so we see that there is a bidirectional almost relationship between the two. Furthermore, in treating with different chemotherapy agents, it's known that the certain tumor cells are going to die, and surrounding cells are going to have an increased mTOR activity as a consequence to try to help them survive. In these circumstances, treating with an mTOR inhibitor like rapamycin, and also further trying to inhibit different functionalities of the tumor via HDR2B in these early nutrient-deprived or especially in early tumorigenic stages, could definitely be a valid dual targeting, two-system targeting method that would definitely be applicable and would definitely need to be currently assessed within preclinical studies before trying to establish anything clinical. Thank you very much for your question.
Thank you very much for the answer.
Thank you so much. As chair